So I'm very honored about this very unique prize and to receive this. This is very, very special to me because it's in principle doing a lot of things. It's not for me, it's also for my whole team, and it shows me that what we're doing, what I'm excited about, is meaningful and valuable because it's evaluated by the scientific community. So as you see here, what I would like to show you quickly is what we're doing is we're exploring how the brain barriers ensure CNS immune privilege. Now, if we look at the brain barriers, and from what Hartmut Weckerle told you when I joined his laboratory until now, we're talking in plural. Because back then, if you just look at a coronal cross-section, we always looked here at the blood-brain barrier. So this is these highly specialized endothelial cells of the microvasculature. What we did not consider so much at the time, that there is the evolutionary older blood-brain barrier, and this is the glia limitans. This is made by astrocytes, and extracellular matrix provides a second barrier. And I will show you later why this is important. Now, more recently, Andrew was alluding to this, a lot of interest has gone to the meninges and the meningeal barriers and the meningeal inflammation in MS. Now here we have the dura mater, and this is important to know, this does not have a blood-brain barrier. Then we have the arachnoid barrier. So these are fibroblasts that make a blood cerebrospinal fluid barrier because in the subarachnoid space, there we have the cerebrospinal fluid. Then again, towards the surface of the brain, we have the pia mater, and then below again the glia limitans. In the center of the CNS, in the ventricles, we have the choroid plexus producing the cerebrospinal fluid, and again there is another barrier, and this is the blood cerebrospinal fluid barrier made by epithelial cells, because here the blood vessels are fenestrated and make no blood-brain barrier. So if you look at the headline, in the meanwhile we have endothelial, epithelial, fibroblasts, and glial brain barriers, and we propose that they ensure together CNS immune privilege. Now, as Hartmut Weckerle explained in the beginning, when I joined his lab, or shortly before, his laboratory and that of Bill Hickey in the United States, they broke dogma. And the dogma was the following. So it was clear from physiology and pharmacology studies that the blood-brain barrier makes a physical barrier because the tight junctions are continuous and circumferential and block the paracellular diffusion. There is no vesicular activity, so again, you have no diffusion through the endothelial cells. Every molecule that needs to get in or out of the CNS is transported by specific molecules and transporters. So the immunologists thought, well, immune cells cannot access or cross this blood-brain barrier because it's a tight barrier. So the immune privilege was considered immune ignorance. Now, Hartmut Weckerle showed the picture. This is the picture everybody shows who was in Hartmut Weckerle's laboratory, because they showed that T cells, if injected into a tail vein or into the circulation of susceptible animals that can develop EAE, so experimental autoimmune encephalomyelitis, that these T cells cross the blood-brain barrier in the absence of neuroinflammation. And this was the dogma break, meaning there is no disease in the CNS, and these activated T cells nevertheless cross the blood-brain barrier. Now, what they do is what we heard before, they induce the animal model, so this is the EAE, but what we can see here with the laminin staining is where these T cells cross. So they cross here the endothelial barrier, and then there is a second barrier, as I told you, the glia limitans, and these red cells reach the parenchyma. If we now look at this cartoon again, you see where the cells exactly extravasate. And this is not like you read in every review in the capillaries. It's at the level of the post-capillary venules. So there you have a multi-step extravasation cascade. These T cells cross the blood-brain barrier, but then they are in a perivascular space. This is not the CNS parenchyma. The neurons are behind the glia limitans and sit behind there. So we and others have shown during the years that there is numerous pathways that T cells can cross at the blood-brain barrier level. Alexander Flügel was actually very prominent, showing with very elegant trafficking studies that also in the leptomeninges across the veins, immune cells can cross into the subarachnoid space. And then together with Federica Salusto, we showed, and then others also showed, that immune cells might enter the CNS via the choroid plexus. Now, because of these findings, there is a large community in neuroimmunology that now say immune privilege of the CNS, that's history. This is totally wrong. And we just turn this around and say, yes, it is history. without the question mark. And I'm very happy that Jörg Bär showed this famous castle. So let's go to Würzburg, because there you see the immune privilege right there. So this is the Marienfestung we have looked at before in Würzburg. And if you look very carefully, it's on the top of the mountain, and it's surrounded by 2 walls. Very good concept to protect the royalties living in the castle, right? So if we transfer this to the CNS, and I think you get the idea, we have the outer wall. This is the blood-brain barrier. And then, unlike in any other organ, there's a second barrier, and this is the glia limitans. Now, as I said, the neurons that communicate by electrical and chemical activity, they need homeostasis. Like the royalties in the castle, they don't want to be disturbed, right? So you have the situation, and then again, the picture fits because you have castle mode guards, and that's what we have in the CNS. That's a population only recently come into focus. These are the border-associated macrophages. They sit in the perivascular and subarachnoid spaces. So they are seeing the cerebrospinal fluid all the time and present whatever is there. Now, like in the medieval times, you know, if there's peace, there is eventually the friend coming by, you know, from the village, and they chit-chat and say, yeah, what's going on, you know. But if there is a danger, you know, this T cell here, so the friend talking to the antigen-presenting cell, will come because there is an infection in the periphery. Now, this activated cell crosses the blood-brain barrier to check with the antigen-presenting cell in these border zones, in the castle mode guard, if that infection has spread all the way into the CNS. If not, then we think that these T cells undergo apoptosis. Remember, you as a person, your neurons will never learn what's going on there because it's in the castle mode. Now, if the T cell does see an antigen, it gets activated, it proliferates. Now, on the outer side, the blood-brain barrier changes. It's opening, right? It has more adhesion molecules. And then eventually these T cells will cross the glia limitans, go into the castle, meaning into the brain, because they have to rescue the host, right? They have to get rid of the infection. And this is when we get sick. Now, in the EAE model, it's very interesting that we have learned that the start of the clinical disease really is coincident with the crossing of the T cells of the glia limitans. Now we have a problem, right? Everybody does intravital microscopy, so do we, and I'm always borrowing this beautiful video from Alexander Flügel, because if we now want to understand if this T cell that is crawling along and eventually leaving the blood vessel here in vivo, where it is, we have a problem, and I hope you see that. It's wiggling around there, it cannot go further, but we have no clue where it is. Because if we look again at the scheme, we don't know if this T cell is now interacting with a cell in the subarachnoid space, doing immune surveillance, if it's squeezed below the pia mater on its way to the CNS, whether it's already in the parenchyma doing damage and it's just squeezed between the cells there, or if it has already exited into the dura mater, a mechanism that was recently suggested by Joni Kipnis. Now, it's very clear we need landmarks. We need more than just seeing the vascular wall. Now, this is what we set out with, and before I start the video, so we used a mouse. That's a VE-Caterian GFP knock-in mouse, and if you just look here, this is the dorsal vein in the spinal cord of a mouse. You can beautifully see in this mouse by 2-photon in vivo imaging the junctions of the endothelial cells. Now, in 2-photon imaging, you see the dura mater because of the collagen in blue. This is the second harmonic generation. And if I now start this video, so we look at the surface, at the meninges of a spinal cord, I think you can very easily appreciate there is more green junctions than just on the vessels. So we saw here everywhere these large cells just below the second harmonic generation, and it looks really a little bit confusing if you look from the top. But if we look from the side, you can see, aha, these layers are at the expected level of the pia mater, and you see it's beyond the vasculature that's filled with this red dye. So what we could show, if I just jump to the next slide, is that indeed these fibroblasts that make the arachnoid and the pia mater, so the border of the subarachnoid space, ceiling and floor, they also express VE-Caterian. So they have adherence junctions that are made by VE-Caterian. Now, this allows us now, if we look and we can do what is called an XY maximal intensity projection imaging, or to look from the side, this is called YZ. So you can appreciate this is the dorsal vein. You have up here the dura mater, below the arachnoid mater in green, then you have a subarachnoid space, and these VE-caterin GFP positive cells also highlight the trabeculae, so the cells around these collagen bundles are also positive. Then we have the blood vessels in the leptomeninges below the pia, and then we get again into the dark, and this is the brain parenchyma or the spinal cord parenchyma. So to just put this side by side, what we have drawn as a cartoon, we can now really image. So this anatomy is correct, and now we can ask the questions whether these borders are really barriers or just borders, and how important they are in regulating immune surveillance and disease. Now, the first experiment we did, we injected tracers. So what you can now see is that if we just look at this one tracer, that the arachnoid and the pia make a border for this large, so this is bovine serum albumin, so it's about 60 kilodalton. So they really form a barrier for this in a healthy animal. And now we can look in neuroinflammation how this changes. And quite surprisingly, just to mention this, the arachnoid barrier does not get leaky in EAE, which we thought quite interesting. Now, what we can also do is, and this is shown here, we can look what the T cells do under conditions of immune surveillance of the CNS. You can appreciate they are still recirculating, they're crawling on the pia. But if we have a neuroinflammatory condition, this looks totally different. First of all, there's way more cells. You can see they're less motile. What you don't see is they're also deep in the parenchyma. Now, if we look at the next video, you can just see this a little bit better. So what we found is that, in principle, the pia mater really does provide a border or a barrier because the T cells recognize it and crawl on it like we have previously observed on the vascular endothelial cells. So it's more or less the same crawling. So, and with the side view, we can see whether it's below or above. Now here you see again a crawling cell, and it's crawling below. So obviously, the glia limitans below does not let it through. So we can observe exactly the behavior, can look now for the molecular mechanisms because we have a precise landmark for this barrier. The next point will then be that we looked how they cross. And exactly like we saw before in the endothelium, they can go through the junctions, which is called paracellular, or what you can see here, this T cell is crossing the PL fibroblast away from the junction, making a pore. So the mechanisms seem to be really similar, like T cell interaction with the vascular endothelium, and that's what we're right now exploring. Now, with this, we are at a level that we can now start to point to where are these immune cells? What is their task? Are they still, interacting to perform immune surveillance? Which layer they're targeting, and where do they go? And we think this is very important for the in vivo imaging. Now, we lack one barrier, and this barrier is the glia limitans. So we have also made a reporter mouse allowing us to look at the glia limitans, and you see this already in red below here. And then have crossed it with our VE-caterin GFP mouse. So now we can look at the vascular, meningeal barriers, and the glia limitans. And if I just start the video, now you're already experts in the mouse spinal cord. So what you can see again is the dura mater. You see the arachnoid below, you see the space. Now you see the red layer. This is the glia limitans, and it's very nicely right below the pia mater. And with this, we have now, in principle, all the layers there, allowing us now to look at injection of tracers, to look how these borders make, in principle, barriers for different tracers or cells. Now, remember the video I showed you from Alexander Flügel? If we now look at this, these videos look way more complicated. Now we have all the colors there, and you can already appreciate this is a cell we found. It's crossing, actually, a vessel, and I just showed this to you. Now we can, with image analysis, really depict exactly where this is. Now, some of you might say, well, Britta, this is nice, but what is the relevance for MS? Well, I want to convince you that this is very important, because in the meanwhile, we still think that the blood-brain barrier breakdown or barrier changes in MS are cause of the neuroinflammation. And I dared to challenge this view as a blood-brain barrier researcher and said, how about the blood-brain barrier contributes actively, independently to the disease? Now, we could not study this before because we had no tools. In the meanwhile, we can make pluripotent stem cells and redifferentiate them into endothelial cells and make in vitro blood-brain barrier models. And we are really pleased that we have a very fruitful collaboration here in Switzerland with Renaud Depakier's lab, who has established these pluripotent stem cells from his MS patients. And we have made in vitro blood-brain barrier models from those and compared those to the blood-brain barrier models of healthy controls. And what we found is that the adherence junctions, over here you have the MS blood-brain barrier models, to the left the healthy controls. What you can appreciate that the tight junctions are interrupted in all the blood-brain barrier models derived from the MS patients. And they have a pro-inflammatory phenotype. So you see this bright red over here. This is all MS patients for ICAM1 and VCAM1. These are the adhesion molecules we and others have shown are important for leukocyte migration into the CNS. And if I just start the videos, you will be able to appreciate in the videos that on the right side, the MS endothelial cells, so the blood-brain barrier endothelial cells, we can now expose to the autologous T cells of the same patients. There's way more interaction, and if they get so dark green, they have really below. And you can see here, this is Hideaki was quantifying these many circles, meaning that the MS blood-brain barrier promotes immune cell trafficking across the barrier. Now, I hope with this short presentation, I have convinced you that these different brain barriers, understanding their anatomy and their function is essential, because they are making a zonation of the CNS, which ensures homeostatic environment in the parenchyma for neuronal function, but at the same time allows for immune surveillance, and this is CNS, immune privilege. And what am I going to do with the prize? Well, we are rediscovering, we heard this today before, we always rediscover. We are rediscovering a pathway from the subarachnoid space, going in the castle picture, right, the secret pathway, to the choroid plexus. And we think this is very important, and we're exploring this for what we see in MS, the periventricular lesions. And this is something we would like to explore with the prize money. With this, I'm at the end. I have to thank my fantastic team, the entire institute, and our collaborators and funders. And I have to thank you as the audience who listened to me, and once more the committee, Hartmut Weckerle, the person who nominated me for the prize. I'm really very honored about this distinct honor and prize. Thank you.